This is part 2 of the Chapter 10 lecture notes, and we left off on this slide, so I wanted to go back over it with you. The stages or the phases in the life of a cell begin with interphase. And so the first stage of interphase is called G1, and that is when the cell grows. It is a phase of cell growth. And the second stage of interphase is called the S stage, and that is when the DNA replicates. Because before any cell divides, it has to double its DNA. It has to make a copy of its DNA so that each new cell that is produced will have an identical copy of the DNA. So that happens during the S stage of interphase. And then there is another growth stage called G2 in interphase. And this cell cycle is drawn as kind of like a pie chart because it represents also the percentage of time on average that the cell spends in each of the stages. And you can see that it spends most of its life in the different stages of interphase. Now, if it's not going to divide, it will enter a stage called G0. Instead of going into mitosis, it will go into a stage called G0 if it's not going to divide again. But any cell that's capable of or that is going to divide again at some point enters the mitotic phase, and that is the cell division part of the cell cycle. So that is when mitosis occurs, which is the division of the nucleus and the separation of the two sets of chromosomes so that each daughter cell, each new cell that forms, called the daughter cell, that each daughter cell will get an identical copy of the DNA from the parent cell or the original cell. And then cytokinesis is the actual splitting in half of the parent cell to form the two daughter cells. The mitotic phase, what's not shown here, is the mitotic phase consists of prophase, metaphase, anaphase, and telophase. And the regions in blue are all representing interphase, and this is the time for normal growth and preparation for cell division. Again, there is G1, S, and G2 phases or stages of interphase. And then mitosis is the phase in red, and that is when the DNA, the nucleus, and the cytoplasm are split and the cell divides in half. And then G0 represents the phase that a cell will go into if it's not going to divide. So the G1 phase stands for the first gap phase. This is the first phase of interphase, or the first stage. There's not a lot of change that's evident, but the cell is growing. And then the S phase stands for synthesis. So this is when the DNA synthesis or DNA replication or duplication occurs. The DNA molecules replicate or form identical copies called sister chromatids. So a chromosome that was once a single rod shape will replicate itself, and the replicated chromosome will be attached to it at a region called the centromere. And each one of these chromosomes here and here are called sister chromatids. So sister chromatids are formed during the S phase. They're joined at a region called the centromere. And the G2 phase, or the second gap phase, is when the organelles are replicated. So not only does the DNA have to replicate and each daughter cell get a copy of the DNA, but all of those organelles that we learned about in chapter 4, the mitochondria, the endoplasmic reticulum, the Golgi bodies, the Golgi apparatus, all of those organelles have to make a copy as well so that the daughter cells will have the same organelles and the same DNA as the parent cell. So those are the stages of interphase. Mitosis or nuclear division is sometimes referred to as karyokinesis. So that is actually a new word for me as well. Every time you change biology textbooks, you learn new things, no matter how long you've been teaching. And this is one that I have always taught it as mitosis, but you can also call mitosis or nuclear division. It can also be called karyokinesis. So the mitotic phase includes mitosis or karyokinesis, and then it includes cytokinesis. And cytokinesis is when the cell actually splits in half. So mitosis is when the nucleus divides. So this represents the parent cell after mitosis is complete. And then this represents the two daughter cells after cytokinesis is complete. You also have an image here to where you can see some of the stages of mitosis. Interphase is here. Prophase I see here, here, and here. Metaphase, where the chromosomes are lined up in the middle. And you can see a cell in anaphase, where the chromosomes are actually being pulled apart or being separated. The chromosome pairs are being separated. Those sister chromatids are being pulled apart to opposite ends of the parent cell. These are the stages of mitosis, and there is a stage included in here that I haven't mentioned yet. It's called prometaphase. So we're going to start with prophase. The very first stage of mitosis or karyokinesis is prophase. During prophase, the chromosomes condense. Remember that DNA, during interphase, DNA took a form called chromatin, and it was very spread out. Now it's going to condense and form these rod-shaped or X-shaped structures called chromosomes. And you can see them. They are visible under the microscope. Another thing that forms during prophase is spindle fibers. Spindle fibers start to emerge from the centrosomes. You may not remember what centrosomes are, but remember a centrosome is composed of two centrioles. So here are your centrosomes and the spindle fibers that are emerging from each centrosome or each pair of centrioles. And the nuclear envelope breaks down. It begins to disappear, and the nucleolus also disappears. So those are the things that happen in prophase. And if you can remember those steps that happen in prophase, telophase will be very easy because it will be the reverse of all of those steps. So the chromosomes condense, spindle fibers emerge from the centrosomes, the nuclear envelope disappears, and the nucleolus disappears. And then you have prometaphase, which of course is just a transition between prophase to metaphase. So in prometaphase, chromosomes continue to condense. Kinetochores appear at the centromeres, and these are structures that the spindle fibers are going to attach to. So this is the most important step in prometaphase, is that the spindle microtubules or spindle fibers attach to the kinetochores or the centromeres of each pair of sister chromatids. And so what they're doing is they're organizing those chromosomes and moving them toward the middle of the cell. And that is when metaphase is reached. When the chromosomes have lined up in the middle at the metaphase plate, that is the stage called metaphase. Anaphase is the stage where the sister chromatids separate. So sister chromatids are pulled toward opposite ends or opposite poles of the cell. Telophase, if you will look back at prophase, telophase is simply the reverse of all the steps in prophase. In telophase, instead of the chromosomes condensing, they actually decondense. Chromosomes are going to form back into, they're going to take the form called chromatin, and they're going to be more spread out. You won't see those rod shapes anymore. The nuclear envelope will reappear and surround the two new sets of chromosomes that have formed, and then the spindle will break down. All of those spindle microtubules or spindle fibers that emerged from the centrosomes, they're going to break down and kind of dissolve into the cytoplasm. And then cytokinesis, which actually occurs along with telophase. These two stages occur together. Cytokinesis is actually the splitting of the cytoplasm. And as you can see in an animal cell, the two cells sort of form an indention, and this indention is called a cleavage furrow. A cleavage furrow. So in animal cells, a cleavage furrow separates the daughter cells. And in plant cells, a new cell plate will form between the two plant cells. So if this is a plant cell and these are the two new sets of chromosomes, you're going to see a very light, under the microscope, a very faint cell plate that will eventually become the cell wall between the two cells. So what you need to know is the differences in plant and animal cells. Mitosis is the same in plant and animal cells, but the cytokinesis in animal cells occurs by the forming of a cleavage furrow and the separation of the two cells. And in plant cells, cytokinesis occurs when a cell plate forms and separates the daughter cells. So now we're going to go through the notes on all of these different phases that we just saw images of. Prophase, the nuclear envelope breaks down. So this is prophase. The nucleolus disappears. The microtubules of the spindle begin to form, so the spindles emerge from the centrosomes, and the centrosomes begin moving to opposite poles of the cell. You do need to remember that centrosomes are composed of two centrioles. In animal cells. Now, plant cells, for some reason, do not have centrioles, but they still have spindle fibers or spindle microtubules that form. They just don't have centrioles. So we're not 100% sure what role the centrioles play, since plant cells don't need them, but spindle microtubules or spindle fibers play the role of helping to organize the chromosomes so they can be pulled apart and distributed into the daughter cells. And let's see, another thing is that I don't really like the way it's worded in the notes. It says at the bottom, sister chromatids coil tighter, aided by condensin proteins. Now, condensin is a term that I've seen on the study guide questions. So condensin proteins aid or help in this process of condensing, like the name suggests. But you do need to understand the chromatin that DNA was in the form called chromatin in interphase. Now the DNA is going to condense and form, that chromatin is going to condense and form chromosomes in the form of sister chromatids. And in prometaphase, the main thing is that you understand that the spindle microtubules are going to attach to a region of the sister chromatids called the kinetochore. So the kinetochore The spindle microtubules or spindle fibers are going to attach to the kinetochore.